Hi again, everyone. Thanks for getting on the Zoom call with me today. You guys are the ones that prompted this review of excitation, contraction, coupling events. This topic comes up in chapters 12 and 13. So my idea for teaching this concept is to first show you how it works in skeletal muscle, and then we just make a few minor adjustments to this theme when we talk about cardiac muscle function. So I want to go back and review some things that we've already covered, but I think it's time well spent if we go ahead and start back with skeletal muscle and a few topics from Unit 2, if we recall a few things. And I've pulled a few notes from your lecture series from both chapters, and just for this talk right now, I've just pulled a few pictures from the old notes that we can really focus on. And make sure you guys have these ideas down before Monday when you possibly get asked some questions about this stuff. So if we go back to our concepts from Unit 2, and we can see here up by number 1, there's a motor neuron, a somatic motor neuron coming in and synapsing, not with another neuron, but in this case, a skeletal muscle. So voluntary control of skeletal muscles. So when the nerve impulse or action potential reaches the end of the axon terminal, shown here as number 1 up at the top of the diagram, there are voltage-operated calcium channels on the axon terminals that open because they're voltage operated, and they are going to allow calcium to rush into the axon, and the calcium inside the axon terminal causes these storage vesicles to fuse with the axon membrane, which is going to lead to exocytosis, so you get the release of stored acetylcholine into the synaptic cleft. And that was something you guys practiced in homework 7. You guys were labeling activities in the synapse. The acetylcholine neurotransmitter diffuses across this fluid-filled synapse, shown here in number 2, and then binds to its acetylcholine receptor on the sarcolemma. These are nicotinic receptors, and they're always excitatory whenever we're looking at somatic motor neurons. So an important distinction to make is that the binding of acetylcholine to its nicotinic receptors initiates an action potential then on the muscle side. So in this expanded view of the neuromuscular junction, we see that the nicotinic receptors are concentrated in this wavy part of the sarcolemma. This is referred to as the motor end plate. So the motor end plate has tons and tons of nicotinic receptors located there in order to catch these acetylcholine molecules as they diffuse across the synapse. From the source of the initial depolarization, this wave of depolarization that's initially happening on the motor end plate will propagate along the surface of the sarcolemma, turning the inner resting memory potential to a depolarized state. And this is indicated here by these hot pink arrows going off to the right. So like you see the wave happening at a stadium where one section at a time is standing up and then they sit down and the next section stands up and sits down. You can think of that as whenever the section is standing up, they're turning positive. And then they sit back down because those channels are going to close down and the cell's going to recover and go back to resting again. So this is how we move an impulse along a membrane. And we covered this idea in Unit 2, just along the axon, and now the muscle is acting similarly by propagating action potential along its surface, just like a neuron did. Okay, so how do we go from the surface, or sarcolemma, down into the muscle fibers? That has to happen with the help of this special structure called the T-tubules. So they're structurally continuous with the sarcolemma and physically linked to the sarcoplasmic reticulum. Muscle fibers have extensive sarcoplasmic reticula in order to make calcium available for contractions. These T-tubules shown here, these structures, conduct the action potentials to the deepest part of the muscle fiber by acting like a conduit or like an elevator shaft, highlighted here in green. So the membrane across the top is continuous. So these little elevator shafts help move the depolarization event into the depths of the muscle. Just feel your biceps. Feel how thick that muscle is. And so the electrical impulses at the surface have to get down into the whole bundle in order for your muscles to work kind of as a team. So when action potentials travel from the surface of the fiber down into the muscle mass, these impulses, or depolarizations, trigger the release of calcium from the adjacent terminal cisternae on the SR. So oftentimes this structural feature between the T-tubule and the SR, we can see the sarcoplasmic reticulum on either side of this green T-tubule. This kind of junction here is called a triad. You probably read about that in your textbook. So along this triad, you can see that there's a physical connection between the green T-tubule and the sarcoplasmic reticulum through two different flavors of voltage-operated channels. The DHP and the ryanodine receptors are both located here, and they're highlighted at the bottom of this picture. You can see in the plasma membrane there's two different colors used to indicate these two different proteins, and they work as a team. They function as voltage-operated calcium channels, and they're going to allow calcium to flood the ICF compartment whenever depolarization comes along. So this activity is how we link or couple excitation that happens at the surface to a functional contraction event. The sarcoplasmic reticulum forms this elaborate, smooth endoplasmic reticulum that mostly runs longitudinally and surrounds each myofibril. These structures are extensive and very well developed in striated muscle types. It functions in the regulation of intracellular calcium levels, which is the link to contraction from the voluntary motor stimulation, which was the initial excitation event. So the way that we couple it is through calcium. Calcium is super important. So let's go ahead and look at another diagram where this is numbered. So this is a really good study tool because you can go through the steps in your book. This is figure 12.8, and look at excitation, contraction, coupling mechanisms in order. So we want to link the electrical signal to the muscle fiber contraction. That is by definition excitation, contraction, coupling. Think of it as a mechanism for turning on or off a muscle contraction. Voluntary muscle control starts with the motor neurons that use acetylcholine, which is always an excitatory effect. Muscle cells, like neurons, are also capable of generating action potentials when the sarcolemma gets depolarized. The motor neuron input causes the myocyte to depolarize at the motor end plate, and depolarization is triggered when the nicotinic receptors open and you have a flood of sodium causing the initial depolarization along the sarcolemma, along the surface. And then we just discussed how from the neuromuscular junction, you can follow the arrows across the top. So I'm at steps 1 and 2 in this diagram. The action potential is going to travel down the T-tubule. So your arrows are going to come down this conduit, and they are going to come in contact directly with the sarcoplasmic reticulum here on either side. And you can see these little spheres indicate calcium storage. So that's why the ICF compartment is always really, really low calcium. It's because the calcium is always sequestered and stored in the SR. So upon triggering through depolarization for release, we can see that here in number 3, the calcium's not going to be in the ICF compartment until it's told to, basically, by the depolarization event. So calcium's going to flood the ICF compartment, and what happens is the coupling event led by calcium is triggered by calcium coming over here to our actual proteins that are responsible for contraction, and this is the actin in blue, and then the myosin here in red. So the thin and the thick filaments are going to interact. And the only way they can interact to actually shorten the sarcomere is measured from Z line to Z line, and that's the actual functional part of the muscle is the sarcomere. But it's packed full of all these proteins. So here in blue, the actin protein has a little friend that calcium likes to pal around with called troponin. And that's this little pink protein interspersed with our blue kind of double-stranded, like a strand of pearls twisted together. That's the actin fiber. And troponin is connected to another long, this is in dark purple here, this long rope-like fiber called troponin. So rope, troponin, rope is kind of in the word troponin. So troponin and tropomyosin work together. So calcium binds troponin. Troponin's connected to tropomyosin, and what happens is the proteins change shape slightly when calcium's around. Troponin's going to pull on tropomyosin, and it's going to expose these binding sites that look like, I don't know, like an olive stuffed with a pimento. You can see these little black dots here. So those are the binding sites. So once those are cleared, you can see over here in step 5, the thick filament, the myosin heads, are going to come over here and bind with actin. And that is called cross-bridge formation. And this can only happen when calcium is around. Then you see the sliding filament. Okay, so these proteins are going to slide past each other. And as long as calcium's around and ATP, we're going to be able to cycle through cross-bridge formation, and myosin's going to ratchet across actin and shorten the sarcomere. And then you see this cyclic pathway here. Okay, so we talk so much about contraction, but really relaxation is just as important as contraction. So what happens is calcium is going to be shuttled back into its sequestered position, so the muscle doesn't go into a cramp or have tetanus, or you get a Charlie horse where the muscle won't relax. It's very painful. So the calcium is going to be pumped through active transport back into the sarcoplasmic reticulum. And what happens then is in step 7, the tropomyosin, one of our 2 regulatory proteins on actin, is gonna kind of sag back down over these binding sites. So these little black dots on our actin where the myosin would normally interact, now they're blocked. So we can't have any interaction. So this is the relaxation phase. When we talk about the heart, I'll do that next. Relaxation in the heart muscle is very important because that is when the blood is going to fill the 4 chambers again. So contraction is going to move the blood through the heart, and then in relaxation, the blood can fill again. So relaxation is just as important as contraction. Okay, so we will review these steps, these numbered steps, and apply this excitation contraction coupling theory. This is an award-winning theory, won the Nobel Prize in the 70s. We'll apply this to cardiomyocytes next. Okay,